Friends, welcome back to my YouTube channel Nastin Padasala. In this video, I'll be sharing you 10 important questions which you can expect for GATE Life Sciences Zoology paper. First question is regarding to embryonic cleavage pattern. So I have given you notes also. If you take echinoderms, amphioxus, they have a holoblastic radial cleavage and they have isolicital egg. Next, annelid mollusk and flatworm, worm VORM. So they have a holoblastic spiral cleavage and they have isolicital egg. Next is tunicates, which have again holoblastic bilateral cleavage and they have isolicital egg. And mammals, nematodes are having a holoblastic rotational cleavage and they have isolicital egg. So isolicital eggs are present in echinoderms, amphioxus, annelids, mollusk, flatworm, tunicate, mammal and nematode. Okay, in echinoderms and amphioxus you can find radial cleavage, and annelids, mollusk and flatworm you can able to find spiral cleavage, and in tunicates you can able to find bilateral cleavage, and in mammals and nematodes you can able to find rotational cleavage. Okay, next in amphibians you can able to find holoblastic radial cleavage and the egg is mesolicital. Mesolicital egg is present only in amphibians. Okay, next is cephalopods, which have meroblastic bilateral cleavage and they have telolicital eggs. Next, fish, reptile, birds, they have meroblastic discoidal cleavage and they have telolicital egg. Next, insects again have a meroblastic cleavage pattern and they have a superficial cleavage pattern and the eggs are centrolicital. So, isolicital eggs are present in mammals, that is human. Mesolicital eggs are present in your amphibian, that is frogs and other salamanders, and telolicital eggs are present in fish, reptile, birds, so it is present in chick, and centrolicital eggs are present in insect, that is Drosophila, which is an insect. So this particular, have a look on this particular page which you can find in Gilbert book. Okay, next there will be a question from cell movements during gastrulation. So here I had given you notes only, so you can check few other questions in the internet. So this is much more important. Invagination means infolding of sheets of epithelium into the embryo. So sea urchin endoderm is formed by invagination. Next is involution, which is an inward movement of expanding outer layer of cell so that it can spread over the internal surface of remaining external cell. Okay. So the amphibian mesoderm is formed by the process called involution. Next, ingression. So if you check the diagram, you can be able to easily correlate the definition. It is a migration of individual cell from the surface into the embryo's interior. And your sea urchin mesoderm, Drosophila neuroblast are formed by this particular cell movement called ingression. Next is delamination, which is a splitting of one cellular sheet into two nearly parallel sheets, and the example is that hypoblast formation in birds and mammals. Next is epiboly, which is an expansion of one shell sheet over the other cell. That is ectoderm formation in amphibian, sea urchin and tunicates are following this particular cell movement called epiboly. So don't confuse between involution and epiboly. Involution is an inward movement of expanding outer layer of cells. So cells will be moving from outer environment to interior. Okay, whereas in epiboly, the cells will be expanding. Expansion of one cellular sheet over the other is called as epiboly. Next, match the therapeutic factor in column 1 with the application. Okay, so if you take factor 8, it is used for hemophilia disease and it is expressed in Chinese hamster ovary cell. And next is streptokinase, it is used for acute myocardial infarction and it is expressed in E. coli. Next is tissue plasminogen activator, again it is also used for acute myocardial infarction and it is expressed both in Chinese hamster ovary cell and E. coli. Next is insulin, which is a humulin, it is used for diabetes and it is expressed in E. coli. Next is erythropoietin, which is used for anemia and it is expressed in mammalian cell line. Next is human growth hormone, which is expressed in E. coli and it is used for those children with this particular hormone deficiency. Next is granulocyte colony factor and granulocyte macrophage colony stimulating factor, which are expressed in E. coli. Next is alpha interferon, which is expressed in E. coli and it is used for leukemia, hepatitis B and hepatitis C and as well as in cancers. And beta interferon is used for multiple sclerosis and it is expressed in both CHO cells and E. coli. Next is gamma interferon is used for chronic glaucomatosis disease, which is expressed in E. coli, and your interleukin 2 is expressed in E. coli and it is used for renal cell sarcoma disease, and hepatitis B is used for preventing your hepatitis B and it is expressed in Saccharomyces cerevisiae. So humulin is used for diabetes, erythropoietin is used for anemia, plasminogen activator is used for myocardial infarction, and this cathepsin K inhibitor. So this cathepsin is actually a cysteine protease enzyme and it is implicated in a process called bone resorption. So bone resorption is nothing but it is a destruction of your bone tissue which will be promoting a bone loss. So what happens when you lose your bone? Ultimately the bone mass, the bone density will start to decrease. This you can find in elderly person group or those with osteoporotic condition or those with postmenopausal condition. So for those you can able to find this bone resorption where there will be severe loss of your bone tissue. So that's why they have discovered the inhibitor to inhibit this particular enzyme. Therefore, we can able to use those inhibitors as a promising therapeutic agent for treatment of disease that is characterized by excessive bone loss that is osteoporosis. Okay, so this cathepsin K inhibitor is used for the disease called osteoporosis and leptin is used for obesity. Okay. Next is Tay Sachs disease is a human genetic disorder which is associated with the defects in which one of the following cellular organelle? This disease occurs due to a defect in this particular lysosome. It is an autosomal recessive disorder that is caused by the mutation in both allele of the gene called hexa. This gene is located in chromosome 15 and this gene code for the alpha subunit of enzyme called beta hexosaminidase A and this particular enzyme is found in lysosome. So you should also know about disease associated with a specific cell organelle. Dynein arms of cells like this Cartagena syndrome is due to a defect in this dynein arm of cilia. So the cilia will not rotate properly. So this syndrome occur. Eye cell disease which is due to a defect in Golgi bodies. That is, it is a genetically inherited lysosome storage disease that is caused by a defective in phosphotransferase enzyme. This enzyme is located in Golgi apparatus. Next is lysosome, that is forms disease. It is a lysosome storage disease. In this disease you can able to find that is you can able to observe there is an absence of this particular acid alpha glucosidase enzyme or this enzyme will be deficient. Okay, next the ribosome, that is Treacher Collins syndrome which is caused by the mutation of gene that will be coding for the protein which is required for assembly and function of polymerases. Okay, so this gene will be responsible for transcribing a gene that make up the cellular organelle called ribosome. So this disease occur, this syndrome occur when there is a mutation in these genes and these genes are responsible for making your transcribing those ribosomal RNA gene which are very sufficient for forming your ribosome. Okay, next this MELS syndrome and labor or heredity aortic neuropathy are due to defect in mitochondria and this both X-linked adrenoleukodystrophy and this neonatal adrenoleukodystrophy and Refsum disease and this Zellweger syndrome are due to defect in peroxisome. Either the peroxisome will be missing or even though it is present it will not be functioned properly. Next, the two homologous genes occurring in different species is called as orthologous. In orthologous, gene will be same but the organism will be different. Whereas in paralogous, the genes will be different but the organism is same. Example of orthologous gene is that insulin gene which is present in your human as well as it can also be present in cow. So the gene is same but the organism are completely different. Okay, in paralog, that is example is alpha globulin gene and beta globulin gene present in mouse. So both the genes are different but the organism is same. And orthologous genes are originate due to the process called speciation and paralogous are due to gene duplication or gene replication even. Next there will be one question coming from this altruism or kin selection or the fancy words in ecology you can able to find ecological behavior. From that definitely one questions will be coming for zoology. Okay, so which of the following is not true? First, altruism reduces the fitness of individual that displays this behavior. Yes, correct. Altruism increases the fitness of other individual in the population. It is also correct. Altruism reduces the fitness of the individual that displays this behavior and at the same time, it increases the fitness of other individual in the population. It is also correct. So, all the three options are correct. The last option is that altruistic behavior helps the individual to escape from the predator. No, nowhere we can able to find this altruistic behavior doesn't help the individual to escape from the predator. Okay. Next is extra embryonic tissue are derived from which one of the following? It is derived from trophoectoderm. Okay, so here you can able to see trophoblast. So what is mean by trophoblast? It is an outermost layer of your blastocyst. Okay, and it is a trophoectoderm and trophoblast. Both are similar only. This a trophoectoderm is the first epithelial tissue that appeared during the mammalian embryogenesis, and this trophoectoderm give rise to an extra embryonic tissue like a placenta or extra embryonic tissue like chorion, amnion, yolk sac, and allantois. Okay. So here I had given derivatives of primary germ layer like from ectoderm, your all nervous tissue, epidermis of skin, epidermal derivatives like hair follicles, sebaceous gland, sweat gland, nails, cornea, lens of eye, and epithelium of oral and nasal cavity and anal canal, your tooth enamel, epithelium of pineal gland, pituitary gland, and adrenal medulla, melanocyte, and cranial bones are derived from ectoderm. Whereas from mesoderm, skeletal muscle, smooth muscle, cardiac muscle, cartilage, bone, and other connective tissue, blood, bone marrow, lymphoid organ, endothelium of blood vessel and lymphatic, so serous of ventral body cavity, fibrous and vascular tunics of eye, synovial membrane of joint cavities, and the organs of urogenital system like ureter, kidney, gonads, and reproductive ducts will be produced from this particular germ layer called mesoderm. From endoderm, epithelium of digestive tract, glandular derivatives of digestive tract like your liver, pancreas, and epithelium of respiratory tract, tonsil, solitary tube, thyroid, parathyroid, thymus gland, and epithelium of urethra, bladder, and an epithelium of reproductive duct and glands are being produced from the endoderm. Okay, so please understand, each will be having a significance, so please remember this particular thing. Cardiac and the cerebral tissue are derived from which of the following germ layer? So cardiac tissue is derived from mesoderm and cerebral tissue is derived from ectoderm. So here you can able to see from ectoderm your neural crest like your melanocyte, cranial nerves, major part of autonomous nerve system, glial cell like nerve cell, adrenal medulla, connective tissue of head, dermis of head and neck, and sensory epithelium of eye, ear, nose, epidermis, and mammalian gland are derived from ectoderm germ layer. Okay, from mesoderm, bone, muscle, connective tissue, urinary system, reproductive system, circulatory system, and dermis. Okay, will be coming from mesoderm. Okay, so dermis of head and neck will be coming from ectoderm, and rest of all will be coming from mesoderm. And from endoderm, gastrointestinal tract, from pharynx to upper rectum, your liver, pancreas, respiratory epithelium, middle ear epithelium, urinary bladder epithelium, thyroid gland, parathyroid gland will be coming from the endoderm. Okay, so cardiac tissue will be coming from mesoderm. So here you can able to see cardiac tissue, and cerebral tissue will be coming from ectoderm. That is all your nervous tissue. Next, which one of the following is the characteristics of deuteriosomes? Okay, so which one of the following is not the characteristics of deuterosome? That's a correct question. Sorry, I typed the question wrong. Which one of the following is not the characteristics of your deuterosome? So first, you should know about uh, protozomes and deuterosomes. Okay, so first protozomes, they are low invertebrate species, and in them mouth occurs first. Okay, next these animal will be having a spiral cleavage, and the coelom will be derived from the mesoderm, and this include octopus, squid, snail, centipedes, millipedes, spider, butterfly, and bee, earthworm, clam, and oyster. Okay, deuterosome go through a process called radial cleavage. So radial, they have a radial symmetric body. That's a correct thing. So this is the correct option. They don't have this particular characteristics. They have only radial symmetry, and they have a well-defined digestive system. At the same time, for deuterosome, in this group of animal, the initial blastocell cavity created by the blastopore develop into anus. So in deuterosome, anus first form. In protozoans, mouth forms first. Okay, so echinoderms, chordata, cephalochordata, urochordata, vertebra like human, birds, amphibian, fish are the example of this particular deuterosome. So hemichordata, everything will be coming under deuterosome. So we have a well-defined digestive system. At the same time, formation of anus occurs first from the blastopore. Okay. Next, which one of the following hormone is incorrectly paired with the function? So, if you check our GATE revision series for both for biotech and life sciences, you can be able to easily answer the question. So, if you check first hormone melatonin, so it is produced from pineal gland and it is function as a biological rhythm. Okay, so that's the correct one. Next is glucagon, which is produced from beta cells of pancreas, and it increases sorry, increases the blood glucose level. Correct. Next is prolactin. So prolactin is produced by anterior pituitary and it stimulates the milk secretion. It is also a correct option. So last thing is calcitone. So calcitone is produced from thyroid gland and it lowers the plasma calcium and phosphate level. But here they had given it increases the blood plasma calcium level. So this is incorrectly paired. At the same time, parathyroid hormone will be increasing the plasma calcium and plasma phosphate level. So have a look on this particular PDF, or you can check the revision series PDF also, video also, it will be very much helpful. The last thing, definitely one diseases question will be coming. So African tick bite fever is caused by Rickettsia species. Yellow fever is caused by flavivirus, microcephaly is caused by Zika virus, and sleeping sickness caused by trypanosoma. Okay, so here you can able to see malaria is caused by plasmodium, leishmaniasis is caused by leishmania, African sleeping sickness is caused by trypanosoma brucei, and Chagas disease is caused by trypanosoma brucei. Malaria, the transmitting agent is mosquito. In leishmaniasis, the transmitting agent is sandfly. In African sleeping sickness, the transmission agent is tsetse fly. And in Chagas disease, the transmitting agent is kissing bug fetus. Next, schistosomiasis, which is caused by this particular parasite, schistosoma, and it is transmitted by water. And filariasis is caused by Wucheria branchofti, and it is transmitted by mosquito. And riven blindness is caused by Onchocerca volvulus, and it is transmitted by black fly. And hookworm infection is caused by Nectitator americans, and ascaris is caused by Ascaris lumbricoides. So here you can able to see the arthropod and the disease transmitted by the arthropod. So mosquito is responsible for causing malaria, filaria, viral encephalitis, and viral fever, and viral hemorrhagic fever like yellow fever, dengue, like that. Housefly is responsible for causing typhoid, typhoid fever, dysentery, diarrhea, helminthic fever, and poliomyelitis. Sandfly is responsible for causing kala-azar, and tsetse fly is responsible for causing sleeping sickness. Louse is responsible for causing this relapsing fever, and rat fly is responsible for causing plague, and black fly is responsible for causing onchocerciasis, which is blindness, and this hard tick is responsible for causing viral encephalitis, and the soft tick is responsible for causing Q fever or query fever, and you can able to see cyclops are responsible for causing fish tapeworm disease. At the same time, it is responsible for causing guinea worm disease. So these are the important list of questions that you can expect for your GATE zoology paper. Okay, so thank you friends, thanks for watching this video.